Okay, so our next speaker, again along the theme of cancer biology, will be Cynthia Davies-Venn. Cynthia is a PhD student at Baylor in the Department of Radiology, and she's going to be talking to us about breast cancer antigens and HER2/neu, and trying to use that as a marker to image breast cancer.
Good morning, everybody. My name is Cynthia Davies-Venn, and the title of my presentation for today is A Novel Peptide Imaging Agent for Detection of Metastases in HER2-Positive Breast Cancer. So currently I'm a second-year graduate student at Baylor College of Medicine in the Translational Biology and Molecular Medicine graduate program. So just a brief overview of my presentation. I'll first give a little bit of background about breast cancer and current diagnostic techniques that are being used. I will then discuss HER2 breast cancer and targeted imaging. And finally, I will talk about my project, which is the development of a peptide imaging agent for HER2 breast cancer. So breast cancer is the second leading cause of death and most common malignancy in women. And this diagram shows the anatomy of the normal human breast, and this here is the fatty tissue surrounding the breast. This is a duct of the breast, and this here is the lobule or milk-producing cells of the breast. So the most common form of breast cancer is actually ductal carcinoma, which occurs in the milk ducts of the breast, which is shown here. And although breast cancer is initially localized to the breast, it can subsequently spread to other sites in a process known as metastasis. And metastatic breast cancer causes about 40,000 deaths each year. And there are no techniques that can specifically and non-invasively identify the presence of metastatic disease. So listed here are some current diagnostic techniques that are used to both diagnose and monitor breast cancer. And the first four techniques listed here are imaging-based techniques, and they're useful techniques in that they're able to identify lesions or areas of biochemical change within the breast that may indicate cancer, but they cannot specifically detect the presence of cancer cells. And the last technique up here, biopsy, in which a portion or the entire tumor is removed for further study by histological analysis, that technique, it can detect the presence of breast cancer cells. However, it's invasive and uncomfortable to the patient. So there is a need for techniques that are non-invasive but can still specifically detect breast cancer. So the subset of breast cancer that my research focuses on is HER2 breast cancer. And HER2 stands for Human Epidermal Growth Factor Receptor 2, which is a transmembrane tyrosine kinase receptor that's been shown to be involved in cell growth as well as differentiation. Now, it's known that about 25% of breast cancers overexpress this HER2 receptor, and the overexpression is associated with a very aggressive disease, increased metastasis, and overall poor outcome for the patients. So in 1998, a humanized antibody that can specifically bind to and block the HER2 receptor was developed and subsequently became FDA approved for use in the treatment of breast cancer. So in addition to treating breast cancer, several research groups have utilized it for what is known as targeted imaging, in which they take the Herceptin antibody and they'll conjugate it to an imaging agent, which is either a radioactive compound or some type of fluorescent dye. And so once they have this conjugate, they can then target cancer cells that express the HER2 receptor. So one problem with using an antibody is that it's quite large, and so it's difficult to clear the body, so it limits its clinical applicability somewhat. So an alternative to using an antibody is to use a peptide. And recently, a 6-amino acid peptide with the sequence KCCYSL was identified by a molecular technique known as phage display, in which a library or large group of peptides are screened to find the ones that will bind most tightly to a particular ligand, which in this case was the Her2 receptor. So this particular peptide was found to bind with a very high affinity to the Her2 receptor. And additionally, the group who identified it, they synthesized a indium-111 conjugate and tested it in an in vivo model. And they found that in vivo it had a high tumor-to-blood ratio as well as a fast clearance rate. So my project involves taking this established peptide and using it to create a dual-labeled imaging agent. So the three components of this imaging agent are the targeting molecule, which is the 6 amino acid sequence peptide, an optical agent, which is a near-infrared dye, and then also a nuclear agent and its associated chelator, which is the copper-64 and the DOTA molecule. So this is the overall sequence of the peptide, and the peptide has been synthesized in the lab by a chemist, Dr. Wei Wang, who I work very closely with. And as the imaging agent has both optical and nuclear components, it's important to note that both optical and nuclear modalities have both their advantages as well as limitations, as you can see listed in the table. So basically, the advantage of having a dual-labeled peptide is that it allows you to basically capitalize on the advantages of each technique while overcoming the limitations. So the hypothesis for my project is that this dual-labeled peptide conjugate will be able to bind specifically to these HER2 overexpressing cells and thus enable non-invasive imaging for breast cancer patients. So my two main research goals at this point will be to conduct in vitro binding studies and to also optimize stability of this peptide conjugate, which currently has a limited stability of only a couple of hours. And then secondly, to test the peptide conjugate in both a primary and metastatic mouse tumor model. So from the preliminary binding studies I have conducted thus far, I've used 2 breast cancer cell lines, which are SKBR3, which overexpresses the Her2 receptor, and this MDA-MB231, which expresses minimal Her2. So these images are from confocal microscope, and the green signal represents cytox green, which is a nuclear stain, and the red represents the peptide. So as you can see in the cells that overexpress Her2, the peptide appears to be binding to the extracellular membrane, which is expected as Her2 is a membrane receptor. And in the minimal Her2 expressing cell line, there was negligible binding seen. So this shows that the peptide does appear to have some specificity for Her2 overexpressing cells. So an additional experiment that was done involved the trastuzumab antibody that I mentioned before, and basically the purpose of that experiment was to determine if this peptide has the same binding site as the trastuzumab antibody. And so what we did was first incubated the 2 different cell lines with the trastuzumab antibody, followed by incubation with the peptide. And so if the binding site was the same, the prediction was that the antibody would be able to block binding of the peptide. So it's a little difficult to see here, but there was a pretty strong signal, so it appeared that the antibody was not able to block the binding of the peptide, which suggests to us that they possibly have different binding sites. So additional in vitro work will be to modify the peptide for stability, as I mentioned previously, to test additional cell lines for peptide binding and localization, and look at different levels of expression of Her2 in those cell lines, and also quantify binding affinity by looking at the dissociation constants. So after the in vitro work is completed, I then plan to look at two tumor models, the first of which is a primary tumor model in which these SKBR3 breast cancer cells expressing the luciferase gene will be inoculated into some immunocompromised mice. And the significance of having the luciferase gene is that it allows for bioluminescence imaging to be used to monitor tumor growth. And so once the peptide is of the correct size, once the tumor is of the correct size, the peptide will be injected intravenously into the animal, which will then allow for imaging. So in order to detect the optical component of the imaging agent, we will use a fluorescence imaging system, which was actually developed by colleagues in the lab. And so basically the way it works is once you inject your agent into the animal, a laser light is shined, which allows fluorescent light to be emitted, which passes through a filter, several lenses, into a specialized camera, which then produces a fluorescence image. And then to detect the nuclear component, we'll be using positron emission tomography, which basically the radionuclide that we're using, copper-64, would undergo decay, producing 2 high-energy electrons, which could then be detected by this sensor here and produce the tomographical image on the computer. So the second model we'll be using is a metastatic model, which was the mouse mammary tumor virus new model, which has been shown to mimic human HER2 overexpressing breast cancer. And in these mice, 100% of them will develop primary mammary tumors, and about 70% will develop metastatic disease to different sites in the body. So the experimental design is to look at early, moderate, and advanced stage of disease to be able to see if the peptide can accurately detect the presence of cancer. And so then the analysis that will be done will be the fluorescence imaging and PET, as I mentioned previously, and that will allow us to look at the peptide's ability to target the tumor and also to look at tumor to background ratio. And additionally, we'll look at biodistribution to assess peptide levels in major organs and to detect if there's any toxicity that's occurring. And finally, we will validate the peptide by using immunohistochemical staining to determine how accurate the peptide is at actually finding or identifying cells that have cancer versus not. So the overall significance of these animal studies is that they will be allow us to assess the safety and feasibility of using this peptide conjugate in vivo. And if successful, we hope that they will be able to provide the preclinical basis for possible future human studies and will help to allow better detection and diagnosis of HER2-positive breast cancer metastases. So that concludes my presentation, and I would like to thank my mentors and advisors, my thesis committee, all the members of my lab, and also the Meyerhoff program for the opportunity to speak today. Thank you.
I was wondering, is it the hope that if you're able to detect metastatic breast cancer earlier, that you can just give more aggressive treatment, or do they can be some kind of other intervening like surgeries? Because usually, once you get to the step of metastatic, if it goes to bone or liver, your options are very limited. So I'm wondering what the usefulness of to detecting it metastatic cancer as opposed to just the rest.
Okay, well, so personalized medicine is actually becoming quite big, and so they're looking at individual, kind of like on a case-by-case basis, and in a lot of cases they have therapies that maybe a certain person would benefit from more than another person, and I think by knowing, I guess, more accurately kind of the course of the disease, or knowing earlier, I guess, the extent of disease a particular person has, they can look more into tailored therapies, which hopefully would be able to help somebody with metastatic disease.
There's a big controversy in breast cancer treatment whether you actually do chemotherapy based on whether you have metastatic disease, and since there are really no good ways to detect it, and going through chemotherapy can be a very debilitating process. If you can actually know that you have it, it's much more worthwhile to do the chemo than if you don't have it and there's no point in it. Other questions?
I was just wondering how specific it is, because you said it's only 6 peptides, and how many other peptides in the body might actually match up and might complement your sequence, but do you know how non-specific binding is?
Right. Well, so from the previous studies that were done by the group that identified the peptide, it appears that it's the nonspecific binding is pretty low. The assay that they used was quite stringent, and so that should, in theory, have removed a lot of the ones that weren't as specific. So this one should be like probably, I mean, as far as binding goes, it should be showing very high affinity for the receptor and probably not binding to other things.
What you do in the mice, were they all female, or did you actually incorporate in these studies with males because males also have breast cancer?
Right. So the plans for the studies involve just females, just because of practical reasons. But it would be something interesting to look at. Yeah.
Do you have any idea how many cells you could detect with your peptide?
Yeah. So that's actually something that we are interested in looking at. I mean, obviously, the lower the detection limit, the more valuable it would be. So that is something that we are thinking about looking into.
And I noticed that some of the in vitro work was done with SKB or MDA lines, which are human lines, but the in vivo work's going to be done with the mouse. So is this a peptide that's conserved between mouse and human?
Yes, it is.
Any other questions?
If it works, is it patentable? Sorry. If it works, is it patentable, the peptide?
The peptide conjugate. Yes, I believe it would be.